Hello everybody, I'm Sandhya Prabhakaran and I'm an applied research scientist at the Department of Integrated Mathematical Oncology, Moffitt Cancer Center, Florida. This is joint work done with Tal Navi and Dana Peer while I was at the Peer Lab at Memorial Sloan Kettering Cancer Center. The title of my work is Sparcle, that stands for Spatial Reassignment of Spots to Cells using Maximum Likelihood Estimation. The images we look over here are what is known as spatial transcriptomics images, or images where single molecules can be imaged at a much fine resolution. An example being MERFISH. So in panel A, we can see an example MERFISH image showing cells as points of green fluorescence. In panel B is the same image but showing the conservative cell segmentation that captures only half of those transcripts shown as blue dots, which accounts to 48% of mRNAs within the cells, and that leaves out more than half of the dangling transcripts shown as pink crosses from downstream analysis. In panel C, you see the same image but now shown with the spatial diversity of transcripts both within and outside the cell segmentation. And each transcript is colored according to the corresponding gene, which in the case of MERFISH was 140. So Sparcle aims to recover these so-called dangling transcripts from multiplexed images. Sparcle does not assume a uniform distribution of transcripts within cells. This is unrealistic given the diversity of transcripts within a cell. And the number of transcripts being so huge, we justify modeling the gene expression as a multivariate Gaussian distribution, and we've checked this assumption using the Lilliefors test as well. For Sparcle, we would require multiplexed FISH-based FOVs, or fields of views, an imperfect region-independent cell segmentation, and the mRNA to gene mapping. The outputs that Sparcle provides would be improved transcript assignment to cells, provides a more realistic per-cell quantification of each gene, a better delineation of cell boundaries, and improved cluster assignments across iterations. So at first, we create a count matrix for all the cells and genes found in the FOVs in order to create a global count matrix. And then you can use a clustering of choice, say a phenograph or a Gaussian mixture model, to identify the cell type heterogeneity. Next, we construct a mock cell per dangling transcript. So here is a zoomed in image to the right which shows a dangling transcript in pink, and around it is a blue circle which actually constitutes the circular window for that mock cell, and every gene that is every mRNA that is present as a cross within this circular window constitutes to the mock cell of that particular dangling transcript. The window size can be determined by the user, and this is an ordered vector. We can also create a weighted mock cell using the inverse distance weighting of each neighboring transcript to the dangling transcript, and this is done using the modified Shepherd's method. So basically, we have for each dangling transcript a mock cell representation of that transcript. Then we use the maximum likelihood estimation for transcript assignment, which is relatively simple and computationally cheap, and you do not need to customize this for different imaging protocols. Once this happens, you can update the global count matrix, cluster, and iterate. And in our analysis, we found that up to 3 iterations is enough for an updated count matrix. This is a graphical abstract showing how Sparcle performs, the input being MERFISH FOVs. And here you can see, due to improper segmentation, how dangling transcripts fall outside the cell segments. This is then the construction of the mock cell around the pink cross, and then how those dangling transcripts are in abstracted as a mock cell can then be assigned to the cells to which they have most similar features. And with that clustering, we can update the count matrix and continue this process. So this is an example of the recovery of transcripts across 3 different Sparcle iterations for one FOV. Each dot is an mRNA dangling transcript, and then the color would show the assignment of that particular transcript to the nearest cell to which it has highly likely similarity. And in panel C, you can see it's all the mRNA assignments from the previous iterations with the unassigned dangling mRNAs removed. And here you see some recovery statistics. Here we show it for all of the FOVs that were used for MERFISH compared with single-seq RNA-seq data. And this would be the single cell RNA-seq data is in yellow, MERFISH alone is in blue, and MERFISH with Sparcle is in pink. So we can see that there is a close similarity of the recovery of the different cell types for MERFISH once we've used Sparkle. And here we can see the fraction of all the transcripts that were assigned back into the count matrix post Sparkle, and those are the regions shown in dark pink. Next, we'll show a very quick example of Sparkle on MERFISH data, both pre and post. Here to the left, you find the cluster-cluster cross-covariance matrices between single-cell RNA-seq and MERFISH clusters. This is pre-Sparkle and post-Sparkle. And we can see that the within cluster covariances are already very dominant because of the dark red color that you see, especially for the non-neuronal cell types. Each column or the row is actually a cell type. And to the right, we see that Sparkle improves the cell type specificity of canonical gene assignments for most of the cell types. So the x-axis would be the same cell types that we saw on the right, and the x-axis would be the canonical genes. So again, over here, we see that there is a strong correlation of the canonical genes with their cell types. For future work, we've planned to modify the maximum likelihood estimation by using a prior distribution per gene for the localization of transcripts, and also implement a maximum a posteriori Bayesian inference procedure. Currently, the code is in Python. We'd like to write this in something more faster, say for example Java. And that was the reason of reaching out to BayLearn to understand if there's any way we can speed up this process, because currently we compute the mock cells or the dangling transcript, which is a very high number, and this can make it computationally prohibitive when you have huge numbers of such dangling transcripts that need to get assigned to cells. So one idea we have is to grid the image and then continue with the mock cell assignments per grid. But again, this is open for discussion, and we're very happy to get any feedback on this particular question. So Sparkle is currently under review, and this is where you can find Sparkle on bioRxiv. These are two ways to reach me, and I'll be very happy to take any suggestions or questions. And I'd also like to acknowledge the funding agencies. Thank you very much for your attention.